Hi, my name is Charles Warden, and I am a Genetics, Genomics, and Bioinformatics PhD student. I'm taking this course in Statistical Genomics, and this presentation is for that course. So there'll be a second section related to some work I'm doing in addition to the requirements for this project. But the main goal is to be able to describe a paper where I can go into more detail on the method. And in particular, I'm going to be describing genomic heritability estimation methods with some emphasis on what in at least one article was called GREML-WGS. I believe GREML-WGS is also okay to say, but when I was preparing for this presentation, I watched a variety of YouTube videos, and I think GREML was the more common pronunciation. So in order to discuss genomic heritability estimation methods, I have to discuss the definition of heritability. So I've got some formulas here, and I have a link provided for a somewhat recent review, but I will also provide a link in the description for this video for a quantitative genetics textbook that I believe is mentioned during part of one of the lectures for this class, in addition to the formula I'm showing here. And so for broad sense heritability, the goal is to study the variation due to genetic effects relative to the total phenotypic variation or the variation in the values for a trait. So narrow sense heritability only looks at the additive effects, which are sometimes called breeding values. But I think perhaps the best way to explain this is to describe what you would be missing. So for this dominant variation, that would be variation that is genetic, but is due to dominant or recessive alleles, so that you could, for example, have an individual with a recessive disease where you didn't actually see the disease in the previous generation. There can also be variation to gene-gene interactions, where I stands for interaction. These are sometimes also called epistatic effects. And those do relate to the broad sense heritability and the total genotypic variation, but those are not going to be captured by the narrow sense heritability. And this is of relevance because for the genomic heritability, you may see this represented as something like H hat representing a estimator and SNP representing single nucleotide polymorphism. And the format is I believe, intentionally similar to narrow sense heritability if the method at most is supposed to capture narrow sense heritability and won't capture other types of genetic variation. So this table was important for making a decision about what to present for this project. And what caught my eye is you have this description of variation from twin studies, and you also have this estimation from what the author called GREML-WGS that matches that estimation. Now, this course has been a great opportunity to learn more about things I've wanted to learn about for a while, both in terms of what's presented in the course and this opportunity to study more about these heritability methods and some other methods. So as pointed out in the last slide, it is important to keep in mind what's being estimated and if it is at most going to capture narrow sense heritability. And my understanding, something that's also important to describe, is that it's possible that the heritability from twin studies could be an overestimation. If that's true, the goal shouldn't really be to get a genomic heritability as close as possible to the twin studies, because then it might actually be an overestimate of true heritability. Nevertheless, as somebody who is trying to learn more about the topic, my individual opinion is that what interests me the most is learning more about this GREML-WGS method and the associated publication. So to discuss GREML-WGS, I first have to describe what is GREML, and this in turn relates to what is REML. So REMLs can stand for either restricted maximum likelihood or residual maximum likelihood, and GREML stands for genomic REML, however you wish to define REML. And so I'm going to provide some links to a couple videos that I think was helpful in order to try to understand what's important about the REML estimation versus what you might call regular maximum likelihood estimation. But my understanding is that it's important that we're trying to solve and characterize variance, and variance in particular can be biased if you use a maximum likelihood method, but a REML estimation is supposed to be unbiased. And it can go through some more details of actually showing you how you can get underestimated variance using likelihood versus GREML, or if I understand correctly, if you go through the calculation by hand, there are certain assumptions about things like the residuals that can be important, and that one can explain why it's sometimes called residual maximum likelihood. But what's important, I believe, for understanding the concept of GREML is that in this early publication, which strictly speaking was, I think, was for height heritability, and there was exactly a method that was given the name of GREML, is they say they use REML to estimate the genomic variability, and the reason they're doing so is to provide an unbiased estimate of genomic variability, because your estimate's biased, and that's the main thing you're trying to study that could be an issue. The other thing that's being introduced is this relationship matrix, which I think currently is more commonly referred to as the genetic relationship matrix, or GRM. But I think it helps us to discuss a little bit more recent paper to help understand that. So this is a paper for genome-wide complex trait analysis, or GCTA, and within it is an implementation of what we call, at least what we now call GREML. And so we've got this equation where you're trying to predict a phenotype, such as height, and you've got some covariates, which I'll describe later, and then this component of the equation is supposed to represent the genetic effect. My understanding from preparing for this course is that if you just use this equation, you'd be considering all the genotypes, and that can be a potential issue with regression if you have more genotypes than you do samples, or more markers than you have samples. So this is converted to something with different dimensions, and in turn, you can see the highlighted text shows that this G follows normal distribution of mean zero and variance A times the genomic variance. And this A is the genetic relationship matrix, which is an estimate of relatedness between the different individuals who each have their own genotypes. And so the point is, this is describing how you can relate the regression model to the GRM and the genetic variance that's being estimated. So I believe one of the next important steps in terms of an implementation of GREML for unrelated individuals is minor allele frequency stratified GREML, or GREML-MS. And I found this to be interesting in the sense that even though different papers could sometimes cite something different for GREML-MS, I believe the most appropriate citation is actually a response to a different article. So this other article describes a method that uses a weighted kinship matrix, and the response to that article is that there are issues that can be improved if you use a weighted GRM, but you can also still use GREML if you stratify the variance based upon their minor allele frequency, so their frequency in the population, separating the GRMs to be calculated separately for, say, rare variants and more common variants. And then the response from the other authors was that they could also reproduce what's being described in the citation for what's now called GREML-MS, but they also got some other issues. And I'm not going to keep discussing this further, except to say that towards the end, I will describe a GitHub repository that I've created, and I've tried to include some references where different methods are used for heritability estimates in the same publication. And I've also tried to provide some references to represent a somewhat diverse set of perspective of what's important to discuss to talk about genomic heritability. What I do think is important for this presentation is that there's been at least one other important revision of implementing GREML for these unrelated individuals. So next step, I believe you'd say, is you could make these bins based upon linkage disequilibrium and the minor allele frequency. And so this is called GREML-LDMS. And so the publication for GREML-LDMS is here. And what I think is nice to present is this other protocol publication, which goes through step-by-step how to run different methods that include GREML. And so for the previous slide, I talked about GREML-MS. So you can see there's these separate bins based upon the minor allele frequency. And then for GREML-LDMS, GREML-LDMS for either of two descriptions either of these two implementations I will briefly describe. You can see that there's 4 quartiles for each minor allele frequency bin. And this ends up being a noticeable number of GRMs that you're going to be calculating. But the idea is that it should be helpful and that there's been evidence that it's helpful. So you can certainly see this large number of different bins for these two implementations. The difference is supposed to be that for this R, the GREML-LDMS-R, there's supposed to be some sort of like average regional LD that's been taken into consideration versus GREML-LDMS-I, where the individual SNP leakage disequilibrium values will be taken into consideration. And then my understanding is that there was another publication between the first GREML-LDMS publication and this protocol paper, and it was they showed the differences in these implementations, and that as described in this publication, I think the idea is that using the individual SNP LD values is the best option, although my understanding is that either way you're going to have some issues like having relatively large standard errors, and that was shown in the earlier table, where even though what I'm referring to as GREML-WGS had the closest estimation to the twin studies, it also had the largest standard error. And without going too much more detail, it seems like that might make some sense if you're having to define all these different GRMs, and I believe make a joint estimate, which you can then have separate estimates that add up to the total estimated genomic heritability. And so now if we go back to the GREML-LDMS publication, we've got this plot where they show some stratified variant groups and the variance explained per group, and at least for height, you can see there's this noticeable increase. Now, what's different for this versus the other paper I'm going to discuss is that there's some amount of imputation being done for variants that are not being directly observed. But either way, there are some similarities in conclusions and plots being used to understand the results. So for this other plot, on the x-axis, you've got the minor allele frequency, and on the y-axis, you've got cumulative contribution to genetic variance. So the highest value is for the most common variance, but it's cumulative. So the total value for all variants has to be 1. And so what they really want you to look at is you've got this sort of trend line here, and then when the dots go above the trend line sufficiently far, that there's supposed to be a relatively greater contribution for that bin versus the other bins. And this appears to be more so true for height than BMI. And then we're going to see what I think is a relatively similar plot to this figure with the GREML-WGS paper. And so now that we're starting to focus more on that GREML-WGS paper, there is a link provided to the GCTA website, which is in general going to be helpful for understanding how to implement the method in different ways. And there are details described for both GREML-LDMS-I and GREML-LDS-R. And this is in particular the GREML-LDMS-I. So you've got this LD score for individual SNPs. And so once you understand the data structure for a certain implementation, you can then go look at the code for this specific paper, and you can tell that they use the GREML-LDMS-I method, which my understanding that's supposed to be the preferred way to run GREML. So I think another component that's important is the overall workflow. And so what you can see is a number of samples are filtered out, and I believe quality control is important for getting good results, and in general is important for studies like this. And you can also see, again, that's important to understand that this method is meant for unrelated individuals. There are other methods that are meant for related individuals, and I believe there are some methods that are designed to work on a mix of unrelated and related individuals, but this is specifically for unrelated individuals. So what you have to do is estimate the relatedness between all samples at least at one point, and if you have sufficiently closely related samples, you essentially have to pick just one of the more related samples, so that what you are up with is a set of samples where no one is more related by some threshold. And then they go even further so that if you have this on the y-axis, the heterogeneity rate, you can see that when they separate by minor allele frequency and then also separate by linkage disequilibrium where it's high when it's red and low when it's a sort of blue-green color, there's a sort of shift in, I guess you'd say, the median for the high LD bins, but they want this to be as similar as possible. And they also want to make sure that you have unrelated individuals. So what they do is they remove any individuals that they are contributing to the sort of the long tails. And so that creates a distribution of heterogeneity that, or I should say heterozygosity that is roughly similar for all of the bins. And I believe there's also some extra QC that's being done to select variance by consideration. And I will try to briefly describe some effect of normalization on your trait of interest as well in the next slide. But the last section actually goes back to the first GREML slide. And so again, we have this formula where the Y is what you want to study, such as height or BMI for various individuals. And then you have this component that's used for modeling the covariates, and in particular describe the covariates we consider are age, sex, and principal components. So that's also based upon the genetics for population structure. And it's being used to try to take in consideration variation that you don't want to count towards your relatedness estimates, because the relatedness estimates in the GRM that are contributing to your heritability estimate. And so just relatively quickly, my understanding is that if you took a direct measure of height and you adjusted for the average between males and females, that would be a decent approximation for a normal distribution, but that's not true for all traits. And in particular, they say that if you look at the BMI, they have this plot of residuals, and it's skewed so that if you do this rank-based inverse normal transformation, I'll provide a link with some more information about details of how this is calculated, then you can get residuals that follow more of a normal distribution. And so that makes it better fit the assumptions of the method. And that's about all I think I'm going to say about that for this presentation, but it seems like it's not making a really huge difference, but it makes some amount of difference for BMI. And so my understanding is the focus is supposed to be on the methods, and so I'm not going to really talk too much about the interpretation, but one thing that I think is important is that they were specifically looking at the impact of using whole genome sequencing data for the heritability estimates. And so for comparison, what they did is they took some relatively more common variants that were present from the HapMap3 project, and essentially they did imputation as if they had a SNP chip that was only designed based upon the HapMap3 SNPs. And what they showed is that by being able to use everything you could call with the whole genome sequencing data, at least after the quality control filters, you could get a higher estimation of the heritability for height and BMI. And they say more specifically that they believe this relates to the ability to include this most rare bin of SNPs. And in particular, if you look at supplemental materials, this most rare bin of SNPs makes up more than half the total number of SNPs. And so I don't know if this is how much is being considered in terms of the difference of size in the different bins in terms of how many variants are within each bin. But now you see something similar to the earlier GREML-LDMS publication in that you've got this cumulative contribution to genetic variants, and you've got a trend line, and you want to see where the points are above the trend line. So these are for the rare bins, and the idea then is that at least for height, based upon where the arrow bars are, you can have a relatively greater contribution from the rare variants. Now, my understanding is that another goal of this project is to be able to describe next steps or additional considerations for the particular paper. So if I had to think of 3 things that I might like to see, then I would first, ideally, like to be able to see this type of analysis in a larger sample size, like maybe like at least double the sample size. And there could be some thoughts about exactly how to do that, but I think that should be possible some way or another. And I would be most interested in seeing what is the heritability estimate and the extent to which the standard errors decrease. And another is that you see this count is for SNPs, and if they strictly mean SNPs and not even things like small insertions and deletions, which you could detect on a SNP chip, then the ability to detect different types of variation from whole genome sequencing data that you couldn't detect from SNP chip data, I think should be something that's worth taking into consideration. And if the method works with relatively rare variants that are SNPs, then hopefully other types of variation could be used to calculate the genetic relatedness matrices that are being used for the heritability estimate. At least I hope that's a feasible thing to do, and I'd be interested to see the effect of that. And then if you do that, and in future studies, because people still say that something needs to be revised, I thought it was interesting how you could go back, look at the assumptions for the method, and find the need to add these stratification strategies. So I'm not sure if I'm correct in assuming that these rarish bin SNPs, the effect of the assumptions has not been characterized as well as these more common SNPs. And so if it's found that any sort of variation from the assumptions can have an effect on the results, hopefully that could be something specific to try to revise the method in the future. And so in the next section, I'm going to try to go through this rather quickly because it's actually all kind of self-initiated extra work that I think is going to be helpful for me to understand the content more, and I'm excited to learn more about the content, but I'm supposed to try to keep this somewhat brief. So essentially, I created a GitHub repository, and one advantage is if you are particularly interested in this GREML method, then I do have some additional information that you can see more about notes I've taken and some additional content that does relate to the first section. And in particular, I believe it actually was a specific part of the project is supposed to be having a like literature review. So I have PDFs uploaded for sort of review of the literature, and I've included the screenshot where there's a lot more content. And so essentially, if you are looking for the GREML information, please know you got to scroll down to this genome-wide heritability estimation notes section. And there's more than I show here, but just know you got to scroll down to get to that. And there's been some changes in what I was going to do, and I had to even figure out what I was going to present on. So what I have here is some data that's being used for reanalysis. And one advantage to this is that if you were to try to do reanalysis of the first main section, a lot of that is controlled access data for humans. And so what I have here is some yeast data, and the runtime, at least for the two crosses that I'm looking at, is really not too bad. And what's very nice is that this publication has raw data, process data, and public code. So that was great for providing a way to help me learn more about these QTL methods. And at least in this context, I believe you have QTL analysis, quantitative trait loci analysis, and then you have the calculation of heritability. But I think it is important to separate these sections because my understanding is that when you have these methods for experimental crosses from a somewhat more, at least somewhat more limited number of parental strains, there are differences in methods versus, for example, the GREML methods where you're looking at unrelated individuals from random sampling. And so, but either way, there is some common ground in the sense that one of the results in this publication was that across all traits, the median contribution of rare variants is 51.7%, despite the fact they constitute only 27.8% of all variants. So regardless of whether you're looking at humans or yeast, there was an emphasis on the importance of rare variants on heritability. And then what I've also done is taken the screenshot of a figure from the paper. And so even though on average they're saying the rare variants are important, the contribution of rare variants varies for different traits. And what is important is that you can go back to code. So here's code that was used to produce this figure. And then you can go back to earlier steps if it's important to be able to understand where the values that are being plotted come from. So I think that's really great, and help to do some additional work on the GitHub repository. And so I do want to at least take a little more time to thank everybody I need to speak that needs to speak knowledge. So in general, you can see a number of different comments or posted for preprints or in post-publication review comment sections. And so I don't always get a response, but you can see that there was multiple responses by the first author for this publication. So I very much appreciate that. And I think that was also a factor in selecting this in addition to the other factors I've described. I very much appreciate the instructor for this course who has provided a lot of information that is understandable and important for topics that I don't have had as much experience in terms of the application. And then I'm not exactly sure what all will be done in the future, but at least so far, I can say that I very much appreciated the comments and questions from my classmates in the class discussions, and hopefully that can continue in various meets. Thank you again. I hope if you are watching this on YouTube, you found this helpful, and I appreciate your time and interest. Bye.